Hello everyone, and thank you for attending today's webinar, Glycomics and Proteomics, a not-so-new frontier in biology. I'm Ellen Williams, Digital Editor at Bioanalysis Zone, and I'll be your host for today's event. Before I introduce you to our speaker, I'd quickly like to cover a few housekeeping items. So you've joined the presentation and will be listening via your computer's audio by default. If you'd prefer to connect to a different speaker, you can adjust your audio settings by clicking the arrow next to the microphone icon at the bottom of the Zoom window. You'll have the opportunity to submit questions to today's presenter by typing into the Q&A tab at the bottom of the Zoom window. You may send in your questions at any time during the presentation. We'll collect these and address them during the live Q&A session at the end of today's webinar. We will also be running poll questions for our audience to answer in today's webinar, and these will appear on your screen in the beginning and end of Clause's presentation. Please let us know your thoughts on today's webinar by tweeting us at bioanalysishzone.com using the hashtag BZWebinars. So we're delighted to have Klaus Lindpaintner from Bruker Scientific presenting for us today. Klaus is the incoming VP of Glycobiology Solutions at Bruker. He's co-authored some 250 scientific papers and holds honorary and adjunct professorships at several academic institutions. He serves on numerous boards, working groups and advisory panels on issues related to the successful implementation of novel technologies in precision medicine. So thank you again for joining us, everyone, and I'll now hand over to Klaus to start today's presentation.
Good morning, good afternoon, good evening, wherever this may find you, and welcome to today's conversation on one of my favorite topics, namely glycobiology. My background is in medicine, in epidemiology, molecular biology, molecular genetics, and I came to glycobiology by way of an area that I have been engaged in for much of my professional career, namely personalized medicine, precision healthcare. And as many of you are aware of, the practice of personalized healthcare relies heavily on understanding in a very differentiated fashion and individualized fashion any one given patient. And to do so, we usually avail ourselves of biomarkers, biomolecules that tell us something very specific about that patient, about their disease. So when it comes to biomarkers, where are we at this point? What is the status quo? Well, it's actually not too terribly encouraging and rather bleak and somewhat depressing, namely that regardless whether you look at protein biomarkers or nucleic acid biomarkers, the performance of these biomarkers with regard to sensitivity and specificity more often than not is fairly modest. So they're either not very sensitive or they're not very specific, or they're not either. And one of the prime examples for that I'm going to get back to in just a little bit is prostate-specific antigen, a very widely used test with a notoriously poor performance. So why is that? Now, one possible answer might lie in the fact that so far we have focused exclusively in our quest for biomarkers on two of the three alphabets or languages of biology, if you will. What do I mean with that? Well, we're all very familiar with nucleic acids and proteins. Genomics, the technology is very well established today, obviously still progressing, and proteomics is really getting there, maturing technology over the last 5 to 10 years. But what we have been blissfully ignorant of for all this time is a third class of information-carrying biomolecules, namely carbohydrates. And carbohydrates have been around for a long, long time, but they have just been blissfully ignored because they have been very, very difficult to study. In fact, the second ever Nobel Prize in chemistry was awarded to Emil Fischer for his work on carbohydrates. And on average, sort of every 7 years since, a Nobel Prize has been awarded in the field, up until the early 70s, when everybody's focus in sort of a tunnel vision zoomed in on genomics and genetics for all the right and wrong reasons, until actually just a year ago, when in 2022, Carolyn Bertozzi once again received a Nobel Prize for her work with bioorthogonal chemistry in glycobiology and glycoproteomics. So what is glycobiology? It is the structure, the biosynthesis, the functional biology of carbohydrates. And when we say carbohydrates, we mean sugars, glycans, saccharides. We'll get into that in the next few slides. Basic concepts of carbohydrates. They're made up of monosaccharides. So those are the smallest units that can no longer be broken down. And in vertebrates, we have a total of 9 different monosaccharides. In microbial species, we have many, many more. But in vertebrates, we just have 9. So that may signal a certain simplicity, but that is belied by the structural and the configurational complexity of these monosaccharides. As there are enantiomers, which are stereoisomers, which are mirror images of themselves. We have diastereomers that are not mirror images. We have epimers that are different just on the chiral center. We have anomers that are stereo or diastereoisomers that differ in their configuration around the anomeric carbon molecule or carbon atom. Those are the alpha and beta ring forms that some of you may have heard of. And then we have conformers, which are the equilibrium of chair and boat form of monosaccharides that, again, some of you may remember from your basic biochemistry classes. Now, these monosaccharides can be combined into oligosaccharides, somewhere between 4 and 12 of these monosaccharides usually, and they form the basis of our glycoproteins and glycolipids. And then we have longer concatenamers, polysaccharides, which are somewhere more than a dozen different individual monosaccharides. They make up very long linear chains and have important structural tissue functions. The glycoconjugates we talk about primarily are glycoproteins. So here we have a protein to which a oligosaccharide is covalently bound. Most of the proteins we're currently studying and that are of therapeutic significance are glycoproteins, such as TPA, such as erythropoietin, luteinizing hormone, all of the immunoglobulins. Then we have glycolipids. Here we have an oligosaccharide covalently bound to a lipid, and these are certainly very important for cell surface structure and cell type as markers and as structural components. And then we have proteoglycans. These are made up of those polysaccharides with a certain amount of protein. And these have very, very large structural diversity, are very large molecules with molecular weights of in the millions of Daltons, so very, very complex molecules that, as I said, are structurally very important in bone and cartilage, for instance. When proteins fold, interestingly or importantly, they fold inward. They kind of crunch up. The opposite happens when glycans are attached. They spread out, and this is why glycans are so important for the cell surface, where they form what we commonly refer to as the glycocalyx, so this furry, woolly surface that covers the entire surface of the cell and very much defines the appearance and the identity of any one cell. So when we talk about glycosylation, the two major classes that we usually refer to are N and O glycosylation, depending on the attachment of the glycan. N-glycans are somewhat larger than O-glycans. They're typically attached to a conserved motif that consists of an asparagine, any one amino acid next to it, and then a serine or threonine. And here we have these attached oligosaccharides. This little cartoon just shows you how these oligosaccharides or how these glycans are usually depicted. There's different ways of depicting them. Some use actually alphanumeric representations, which are very, very complex and very difficult to understand. Most people today use these symbols, squares and circles and triangles of different colors to depict these glycans. And then we have O-glycans, which are usually smaller glycans, but generally, if and when they occur many, many more on any given protein, because They don't rely on any particular motif, but can attach to any serine and threonine. They're typical, for instance, for much of the mucin molecules that cover our cell surfaces as well. So here's once again just a quick little cartoon again of how these different glycoconjugates appear on the cell surface as glycosphingolipids, as glycoproteins, and then as these very, very large proteoglycans or glycosaminoglycans and some other structures that we're not going to get into today. Suffice it to say, glycans add dramatic complexity to the proteins and the proteome. And if we look at the building blocks and how they are being put together, that explains this complexity. So while we actually have more building blocks in proteins, 20 versus 9, the way they can be put together is much more diverse when it comes to glycans. 3 to 4 different linkage sites, 2 instead of 1 linkage configuration, 6 to 8 to even more homodimer structures. And then, of course, a linear structure on the protein and a very complex branched or linear as well structure on the glycan level. So if you follow me in a little thought experiment, if we look at how many different peptides can we construct out of 6 different amino acids, 3 different amino acids, well, we can come up with 6 different structures, and here they are. If we take 3 different glycans with all of their conformational individualities, we can arrive with those 3 building blocks at over 1,000 structures. So add to that then two additional layers of heterogeneity that we refer to either as macroheterogeneity. This refers to the fact whether or not all or any or which or none of the motif present asparagines in any one glycoprotein do carry glycans. So this is the occupancy, as it also is referred to. And then microheterogeneity, which refers to the actual nature of the sugar molecule, the glycan, the oligosaccharide, that is attached to any one of those asparagine residues. And so if we carry this thought experiment a little bit further, if you imagine a protein that has 3 different glycosylation sites, and on 2 of those glycosylation sites, we find a total of possible 5 different glycans, and on 1 we find 7 different glycans. Now, the way these can be composed, these 3 sites with these 5, 7, and 5 glycans, gives us a total of 175 possible glycoforms of this one protein, and that certainly completely ignores the added complexity that would be imposed on that if we were also to take into account the configurational and conformational isoforms of all of these different glycans. Add to that then, to that level of qualitative complexity, a quantitative complexity, namely that any one of those 175 different glycoforms can occur more or less abundant at a dynamic range of about 3 to 4 orders of magnitude, that adds on to the already well-known dynamic range of about 10 to the 12 or so that Lee Anderson has taught us about. Then you can just imagine how complex this layer of biology is and how information rich at the same time it also is. So putting all of this together, as I mentioned, genes and proteins are simple because they are synthesized on a linear template. We don't have a template for glycans. It's about 500 different enzymes that in very, very complex reactions in different compartments of the cell make up the branched or linear structure of these glycans. Many, many more building blocks, of course. The complexity is much, much higher theoretically at least. And then again, we're adding an additional layer of dynamic range to each protein by breaking it down to its glycoisoforms. So once we combine glycomics, if you will, and proteomics, we arrive at an incredibly large and rich ocean of information and complexity. And the downside, of course, is that we need to decipher that complexity and that information. And that information is important because, very importantly, it is not static. It's quite dynamic. So as physiology changes, as a healthy cell transforms to a diseased cell, glycans, protein glycosylation can change dramatically, and that's been known for a long time. And in as much as this is, of course, very informative on the tissue level, some of these proteins, glycoproteins, then get shed into the circulation, become secreted glycoproteins that can potentially be picked up as uh, regular blood tests in the sense of a liquid biopsy, which of course makes it then potentially much more approachable, much more accessible, much more actionable from a medical and a clinical standpoint. So this is really where the promise of these glycoproteomics, glycopeptidomics biomarkers lies. Um, once we arrive at a deeper understanding of this entire layer of biology, it's bound to dramatically influence, improve the way we can find new biomarkers, new information about patients that can go into how they are treated and how they are being dealt with. There's another aspect of glycobiology that's, I think, is very important, namely the way we look at protein diagnostics today is we measure their level, we measure their abundance. And if a protein is expressed at the same level in a healthy and diseased state, it's non-informative, obviously, and we ignore it. Well, we may be losing a lot of information by doing so, because, as we by now know, this one protein can easily be present in at least two different glycoforms or even more. And this cartoon depicts for any given protein here two different isoforms. Here we have a triantennary glycan and a biantennary. Here we have a tetraantennary and another tetraantennary. And let's just assume that in the healthy state, 20% of that protein is present as form A and 80% as form B. And in the diseased state, despite the fact that the overall level is identical, now the proportions have shifted, and 60% instead of 20 is now form A and 40% is form B. And that obviously potentially is important and informative. And yet, if we use any of the currently used affinity-based, in other words, antibodies, technologies such as ELISAs, we will never see that. We will never discover that, because antibodies, with rare exceptions, are not specific to glycoisoforms. And so we will have the same test result, but in fact, if we could drill down to that lower level, we would actually gain a lot of additional information. So this is really the promise in many ways of adding glycobiological interrogation and investigation to what we're doing already in terms of proteomics and protein diagnostics. Let me just briefly touch on the terminology, because it tends to be a bit confusing. So there's glycomics. And while some people, when they talk about glycomics, they mean everything that has to do with carbohydrate biology, such as individual mono or oligosaccharides, glycoproteins, glycopeptides, glycolipids. Others refer to glycomics more specifically as those glycans that can be released from their conjugate molecules. So what these investigators refer to is a situation where you take an enzyme, such as PNGase F, digest the glycoprotein or glycopeptide with that, and then separate the glycans from the protein and just measure the glycans. You can do that in a targeted fashion by purifying the protein first. And so you will then know what are the glycans that are present on that protein in the healthy state, in the disease state. You won't know any longer from which glycosylation site any one of those glycans come. Or you can do a global experiment where you use a complex protein mixture. Again, you shed the glycans by using this enzyme. You find out what are all of the glycans that are present in this particular sample. You will lose now not only the glycoside information, but of course also the protein information. So you're giving up a lot of information, but for some particular applications, that may still be informative and may be fine. And a lot of work is being done on that level. And then there's the more, if you will, sophisticated level of glycoproteomics, glycopeptidomics, where you actually capture all of the information. What is the protein? What is the glycopeptide and the glycosylation site? And what is the glycan? And that, of course, is the richest source of information, of course, also the one that is most difficult to attain and to arrive at. So I've probably bored you already now long enough with all of these basic concepts of glycans and carbohydrate biology. Is it meaningful? Is it important? Well, in the next few slides, I hope to convince you that yes, it is. And in fact, one of my pet peeves in glycobiology is the term that many people refer to when they talk about glycans, namely as decorating proteins. And nothing actually can be more wrong than that particular description, because glycans are anything but a decoration. They are structurally and functionally highly relevant. That isn't necessarily evident if you look at a 2D representation, where here we have the peptide backbone and here the little sugar molecule sticking out. But if you look at the 3D representation of this molecule, then you will see that the glycans actually very much define the appearance and the identity of this protein, can either shield it from and masquerade it as self from immune recognition, or at the same time sometimes can be highly immunogenic themselves. It's important to recognize that, particularly with regard to uh, biologically important proteins, up to 70% of them are glycosylated. So by far the most common kind of post-translational modification. Glycans often can be larger than the actual protein molecule that they are attached to itself. They can and more diverse. They can therefore dramatically alter, define, supersede that protein's function, their parent protein. It's important to recognize that 2% of the mammalian genome is dedicated to glycosylation. So clearly, if nature does that, it's doing it for a reason. That is not just some decoration. And what we do know, even though we're still at the beginning of understanding of all of this, we do know that glycosylation and glycoproteins are extremely important for cell proliferation, for all kinds of biological functions as differentiation, migration, cell-cell recognition. The very basis of life of all of us is based on the recognition or on the docking of a sperm to an egg cell with the help of glycans that recognize each other. And of course, host-pathogen interaction, all of viral disease is heavily dependent on the glycan, the glycosylation of these viruses. And then last but not least, it's important to recognize that almost all currently used FDA-approved protein-based diagnostics, and certainly all biotherapeutics or biologicals, are glycosylated proteins. The best way to demonstrate how important glycosylation is the fact that it forms the basis of our ABO blood group, something that was discovered by Karl Landsteiner in 1900, and that we now understand is based on very, very tiny differences between a particular glycan on the surface of the red blood cells that differentiate these red blood cells in either blood group A, blood group B, blood group O, or blood group AB, and that certainly can make, as we all know, the difference between life and death with regard to either a life-saving transfusion or a potentially lethal transfusion reaction. Glycans define the protein's true appearance, as I've mentioned. Here is an example that we can't leave out today when we talk about glycobiology, namely the S-spike protein of the COVID virus, which looks rather simple if we just look at the protein, but very, very different when we look at its actual appearance with all of the glycosylation residues that are attached to it. And you can just imagine that if that were not the case, this molecule would be in a very different place in terms of docking on its ACE receptor, and potentially the course of the disease would be possibly dramatically altered. The metaphor that's commonly used for this is the forest. So when we just look at proteins, we're looking at a biology textbook image of barren tree trunks in a post forest fire forest. That does not at all represent the reality, namely the very rich and lush tropical forest canopy that's made up by the glycans that are attached to the stems of those proteins. And Ajit Varki, one of the leading thinkers in glycobiology, the lead author of the most important textbook of glycobiology, says that evolution has failed to generate any living cell that isn't reliant on surface glycosylation, or that is devoid of surface glycosylation. So another nice example how important glycosylation is, a molecule that I've already made reference to, namely the prostate-specific antigen. This, as you all know, is a screening test for prostate cancer that's being used millions of times a year on men at a somewhat more advanced age. And it's recognized yet as a notoriously poor test that has a low sensitivity, low specificity. And it may just be because when we measure PSA, we ignore the fact that this is not just one simple molecule, but that it is actually made up of a number of different glycosylation forms, some of which, namely the biantennary form, are occurring normally in healthy states or in benign prostatic hypertrophy, whereas others are typical, a tetra or triantennary form for prostate cancer. And if we use the conventional immuno-based assays such as an ELISA to measure PSA, all we're looking at is the peptide backbone, and we're throwing all of these oranges and plums and apples that are representing, if you will, these glycoisoforms in one bucket, and we end up with a performance of this test which, as I said, is notoriously poor, an overall accuracy of about 65%. And for those of you who are not familiar with these receiver operating characteristic curves, if the curve is basically on the diagonal, that means a sensitivity specificity of 50%, so a toss of the coin, a useless test. The further up this curve goes into the corner of this graphic, the better the test is. If it's really up in a corner, it's 100% sensitive, 100% specific. So this is a pretty poor test. Now, if we were to be able to differentiate between these submolecular forms of PSA, then presumably we could do better. And we can differentiate with that with technologies that go further than just antibody technology, namely either nucleic magnetic resonance or mass spectrometry. And if we use these technologies, what we find actually is a performance that is dramatically better. So now, if we're basing our distinction or our differentiation on the glycoisoforms, we get to an accuracy of 95% and a test that very nicely differentiates between benign prostatic hypertrophy and individuals who are at high risk for prostate cancer or who already have prostate cancer. So very dramatic opportunity here that so far has not been taken advantage of, simply again because these mass spectrometers are not the easiest and not the simplest instruments to run and to deploy on a day-to-day clinic basis. Another example for how important how important glycosylation is therapeutic antibodies. I already made reference to that. Therapeutic antibodies on their Fc domain have one particular glycosylation site. It's asparagine 297, which, as we now know, can dramatically affect monoclonal antibody potency. So this is an example of a CD20 antibody, rituximab, that's been around for a long, long time in a number of different indications, non-Hodgkin's lymphoma, CLL, various autoimmune diseases as well. A drug that's manufactured and marketed by Roche. Now, a small company called Glycart outside of Zurich took this antibody and modified that glycan. It deglycosylated and desialylated it and came up with a new molecule that they termed GA101 and that finally reached the market with the generic name of obinutuzumab. And the performance of this particular therapeutic antibody that differs from the original one only in the glycosylation site turned out to be literally twice as good as that of the original molecule. So the median progression-free survival, I think this is a study in CLL, in chronic lymphocytic leukemia, was doubled when that deglycosylated or partially deglycosylated antibody was used as compared to the glycosylated antibody. So very, very dramatic impact, and clearly testimonial to how important glycosylation is. And there is, in fact, a large and ever-growing body of literature about changes in glycosylation as they happen in a variety of diseases, specifically, of course, in cancer, to some extent because cancer is always most easily studied because we do get access to the tissue. And there's abundant evidence that protein glycosylation changes all over the place, oftentimes increased sialylation, increased fucosylation. We know that, but what we don't know is what is the meaning of it and to what extent is it actually contributing to cancer behavior or cancer growth. So there's a lot of work still to be done. And again, given where we are today, where we're starting to have the tools to actually drill into this, there's a lot of potentially very, very important information that we're just about to start digging into or digging through. There's a number of other disease indications such as autoimmune diseases, inflammatory diseases, infectious diseases, neurodegenerative diseases, where again, there's plenty of evidence for changes in glycosylation, for aberrant glycosylation, and we just need to do more research to understand what this is all about, how it could be applied either as biomarkers or as novel therapeutic targets to what this is all about, namely treating our patients. So what are the challenges if there are all these promises? Why haven't we gotten further of this field? Well, one of the challenges is that glycobiology is still a niche science. In as much as everyone who is in the biomedical field has at some point heard about glycobiology or glycosylation, that's usually where it stops, and they don't know any much more, and it's usually perceived as a very, very difficult field to study. So there are these fairly dramatic asymmetries across the different domains of molecular research when we go from genomics to transcriptomics to proteomics to metabolomics to lipidomics, finally to glycomics. What we find is a decreasing knowledge base. Clearly, this has been studied much more deeply than any of the others. In some way logical, but on another level, illogical level of funding. Of course, the more knowledge that's being generated, the more grant proposals are being written, the higher the funding ultimately is. The number of scientists is largest in genetics and then decreasing in other spheres. And the scientific popularity, the overall recognition of the field clearly is much higher or goes down along this cascade. And yet, arguably at least, when we look at information content, it's the opposite. Because this is so complex, at least theoretically, there is more information present in the glycome than in any of these other domains of biology. So going back to JFK, who said we choose to go to the moon not because it's easy, but because it's hard. We at Bruker have chosen to do glycomics not because it's easy, and not necessarily because it's hard. We just accept that it's hard, but because it is so important. And unless we figure out how to deal with the difficulty, we will never get there. And that is our mission now, to make this easier, make this more accessible. So moving back to those challenges, again, as a result of this, the glycobiology community is still relatively small. It's very dispersed. And as my mentor Ajit Varki says, and he states this, he cites this statistic, there's basically one glycobiologist for every 250 or 300 molecular biologists. I don't know where the statistic actually comes from, but he's probably not too far off. There are modest resources and modest support for networking, for collaborations among the community and beyond into clinical medicine that are currently being expended. The infrastructure, the technology, really up until now has been not as strong as we would have liked it to be. But like I have been referring to before, I think we are now at an inflection point where we're starting to cross the threshold, and where we can apply technology that is powerful enough to actually give us new insights. And so we're beginning to move from what we up until now could at best do, namely decipher the composition. So what are the actual monosaccharides that are in any one oligosaccharides, to moving to structure, to actually deciphering what is exactly the branched configuration or structure of any one glycan. This is made possible by newer features of fragmentation engines. So this is mass spectrometry technology with the appropriate throughput that also allows us to apply this to the kind of clinical studies that rely on looking at hundreds and thousands of patients. Oftentimes, a single run on a mass spectrometer for a single sample takes an hour. Now, unless we can speed this up, we'll never be able to do thousands of patients. So that's very, very important. We are now working hard on developing powerful, fast, both qualitative and quantitative analytical softwares. That's an area of great need in the field, just as is the availability of the kinds of comprehensive databases and libraries for glycans or glycosylation pathways that we have had for a long time in nucleic acids and in proteins. And then in addition, we still need to work on finding better technologies, physicochemistries to enrich for the glycan moieties among all of the proteins to actually access them, some of which are, as I mentioned, at very, very low abundance. So we need to enrich, we need to purify them. There currently is a transition, I believe, of glycobiology into mainstream molecule, and the fact that all these therapeutic antibodies are glycosylated and that glycosylation is so important for them clearly is only the tip of the iceberg. There is still a huge lack of awareness among both glycobiology and clinical audiences that we need to do our very best to breach and to make a thing of the past. And so in so many ways, and this is again a term coined by Ajit Varki, we're faced with glycophobia, the irrational fear of getting involved in the understanding of biological processes. And as I said, many, if not most, of biological processes rely on glycans, of those processes in which glycans play a major role. So let's not be threatened, or let's not be intimidated by that. And at Bruker, one of the things that we have committed to is actually making it easier to the community to do glycobiology by putting together what we are looking at viewing as sort of a soup to nuts solution suite, incorporating hardware, software, wetware, application development that will make it more accessible to the non-super glycobiology specialist and allow the field to have the kind of impact that all of this is ultimately all about, namely to change the human condition and to improve the lives of our patients, to provide them new treatments and new therapies. So the field is at an inflection point. These are projections from marketing analysts that are predicting that there's going to be a exponential increase in activity, and also, of course, from our perspective, in the number of instruments, the kits, the reagents, the enzymes that are being used and being applied to this particular field, with expected compound annual growth rates of 10 to 12 to 15% over the next 10 years. So clearly we recognize that this is something that is just about to take off, and that is going to be more and more important as we move forward. And therefore we are continuing, and we're committed to continuing to pushing the frontiers and making progress and committing to innovation as an essential fact of life, because as an old African proverb goes, if you prepare for tomorrow, tomorrow will come. If you don't prepare for tomorrow, tomorrow will come just as well. So this now brings me to the end of my remarks. Thank you so much for being with us today and for your attention. I hope this was helpful. I hope I've been able to communicate how important, despite its up until now fairly fair obscurity, glycobiology actually is as a field of research and as a field of potential biomedical importance. I would encourage you to reach out to me if you have any additional questions. My email address is here on the slide. And we're also always interested at Bruker to potentially collaborate with scientists, with investigators, specifically myself in the field of glycobiology. So please feel free to reach out. I'm now going to hand over back to our moderator for a brief Q&A session. And again, thank you so much for your attention and for being with us today. Goodbye.
Lovely. Thank you so much for that presentation, Klaus. So we're now going to move into the Q&A portion of today's webinar. So if you do have any questions for Klaus, then please do continue to submit those questions using the Q&A button at the bottom of your screen. So we're going to start addressing some of your questions in a minute. But firstly, Klaus, what are your thoughts on some of the poll results that we were collecting throughout this session?
So I'm on the one hand encouraged, and on the other hand not terribly surprised. I'm encouraged that most people actually felt that doing glycoproteomics rather than just glycan analysis is where the true value lies, and that clearly is where we gain or gather most of the information on the protein, the glycosylation site, and the glycan, rather than just what the glycans are as with released glycans. And then with regard to where the challenges lie, not surprisingly, a lot of people felt that the informatics, the analytical software that we currently have is not where it needs to be. So again, this is something that we're all painfully aware of. And there's a lot of work being done on that, but clearly this is an area that's very key. So yeah, those are two of the things that stuck out to me.
Great, thank you. It was lovely to hear your commentary on those as well. So the first question that we've got for you from our audience. So somebody's asking here, so can I use other methods than MS to analyze glycosylation?
There are other methods out there. There's a class of proteins called lectins that actually specifically recognize glycans, and they've been used sort of in the sense of an antibody recognizing a protein. The problem is that they're not terribly specific. They tend to be difficult to characterize, and they only cover a certain spectrum of all the different glycans. So it's something that, yes, has been used and is being used by folks. And there's actually a huge potential there. These are all proteins or peptides that come from natural substances. And what we still need are larger libraries of lectins, particularly since, if we could find a lectin that is as specific as a mass spectrum for presumably a clinical test, then that would certainly be the way to go. It would be a hell of a lot easier just from a practicability standpoint than using a mass spectrometer, which is a very expensive, very complicated, and to some extent still rather temperamental instrument that needs a lot of tender loving care. So yes.
Great, thank you. So our next question here, and definitely something that I've also wondered, is protein glycosylation different in patients with diabetes?
It's a very interesting question, and there's actually not a lot of data on that.
There is also the difference between glycosylation and glycation. So all of you, or many of you, may know about hemoglobin A1C, which is a marker for a long-term marker, much better, much more stable than blood glucose for how well a diabetic patient is treated. And depending on how high or low their sort of average blood sugar over a period of time is, the higher the glycated portion of this hemoglobin A1C is. What glycated means, it's sugars that are associated with a particular protein, but not covalently bound. So this is something that just happens because the protein is exposed to a high concentration of sugar, but not by virtue of the action of enzymes. So those are two different things. What we don't quite know at this point, and it's something that I'm actually quite interested in, but what we would need is some very specific cohorts and patient groups, is to look at whether any one of a scope of different circulating glycoproteins look different in patients that are diabetic versus non-diabetic, or diabetic or well controlled versus not well controlled. So there's lots of work to be done. Thanks.
Great, thank you. So another question here from our audience. So are glycosylation patterns heritable?
Very interesting question. In as much as at some point, glycosylation patterns depend on the presence of certain glycosidases and glycosyltransferases enzymes. They are heritable, and there's actually a class of diseases called congenital disorders of glycosylation, CDGs, very rare diseases where one or the other of these enzymes are missing, and these can be very dramatic syndromes or actually very subclinical syndromes. These are clearly heritable. Whereas what happens actually on a day-to-day level, if you will, to the glycosylation state of any one protein, that is something that depends a whole lot on environmental factors, on lifestyle factors, on diseases that may or may not occur. To what extent these things can be inherited, actually, if you will, in the sense of an epigenetic inheritance, so not a germ cell inheritance, but an epigenetic inheritance directly from mother to child, such as mitochondrial changes can be transferred directly. That's an area of current investigation, a very interesting one. And the feeling is that, yes, some of these glycosylation changes, very similar to DNA methylation, can actually be transmitted from mother to child without any change in the germline DNA.
Oh, that's incredibly interesting. So another question here that we have for you, Klaus. Do you think, in your opinion, is protein glycosylation important for biotherapeutics?
Oh, absolutely. I mean, I showed this one example, the famous asparagine. This is the canonical N-glycosylation site where you've got an asparagine, then any other amino acids except for proline, and then either a serine or a threonine. That the Fc fragment of any one IgG antibody has a glycosylation site there at this asparagine 297. And as I've shown in this one example with the two monoclonal antibody drugs, depending on what that glycan looks like at this particular site, you may have a very potent biotherapeutic or a much less potent biotherapeutic. That's what people have been focusing on right now. That's very well understood. There's actually a whole lot of other glycosylation sites on the FAB fragment of therapeutic antibodies that so far have not really been looked at, not very deeply investigated. But again, presumably, since nature bothers to make those glycosylations, they probably do have some effect and are of some importance. Again, there's a lot of work still to be done. What we know today, what is very, very clear, is that lots and lots of things change with regard to glycosylation in health and disease. What it actually means, aside from potentially, the most obvious use would be biomarkers, as long as any one glycosylation change is robustly associated with a particular disease or diagnosis. To what extent these could also be new therapeutic targets, that's not quite known, even though there's a number of small companies currently active that are specifically targeting glycosylation changes or glycobiology with regard to novel therapeutics. Definitely, yeah.
Great, thank you. So we've got a couple of questions in one for you here. So are there various enrichment strategies available to enrich for certain glycans? And is enrichment in general needed to perform glycoproteomics?
So you can do glycoproteomics just on plasma or on cell lysates. But clearly, in as much as the power of the mass spectrometer is its specificity, vastly more specific than any antibody, which is always somewhat promiscuous. Its downfall, if you will, I don't see it as a downfall, but its limitation is usually its sensitivity. It's not, by far not as sensitive as an immunoassay, or certainly not as a radioimmunoassay. So yes, while you can see about roughly 70 or so circulating glycoproteins without any kind of enrichment, you can go down, you see down to somewhere around 200 nanomolar. Anything that's lower than that, and then many of the interesting proteins, such as all of the cytokines and the chemokines and many of the better known cancer biomarkers, such as PSA, such as CEA or CA125, they're all in the 200 or lower picomolar range. So you don't see those if you're just not using any kind of enrichment. There's a whole slew of different enrichment technologies that are being used. The best known probably is something called HILIC. And it's basically a chromatographic enrichment where you are getting rid of all of the, what you do is you take your protein, you go through a tryptic digest, you end up with a lot of peptides that do not carry any glycans, and then a few of the peptides that happen to have one of those asparagine residues that carry the glycan. The glycan is then bound to this particular resin, and then you elute it, and that's how you concentrate or enrich for it. There's a whole many different other different ways of doing this. There's still quite a bit of controversy about what is the best way of doing it, but yes, in principle, people will very commonly enrich for glycoproteins.
Great, thank you. And I think we've got time for maybe one final question. So one of our audience members here is saying, so glycosylation on asparagine at different sites results in different isoforms. So how do we separate those proteoforms or isoforms?
Again, by mass spectrometer and mass spectrometer analysis, we can find exactly what peptides occur or what glycopeptides occur with regard to the glycans that are associated with a particular peptide. So we break down, once again, we break down the protein with trypsin. We end up with our selection of glycopeptides. Those are the pieces or the peptides out of this whole protein that actually carry this canonical asparagine. They now carry any one of a number or maybe only a single glycan. And on the mass spectrometer, we can resolve all of this. We can find out that of that particular glycopeptide, 30% carry a glycan A, 20% carry glycan B, and 50% carry glycan C. That is certainly doable. It's, again, the quantitative aspect of all of this is still a challenge, particularly also because internal standards calibrators are very far and few between. They're very difficult to come by. Synthesizing internal standards or calibrators for glycopeptides is challenging. It takes a long time. It is very expensive, and you can't make just every glycopeptide. So that's still an area of ongoing exploration and work. As I said, we're at the inflection point, but I believe that where we are right now is very, very promising. I think in glycoproteomics, we are today where we were with proteomics maybe 5-10 years ago, and with genomics 15-20 years ago. We still have a lot of work to do, but we now are getting the tools that are needed to actually address this very, very challenging area. And like I said, there's no free lunch. Or as people say, there's no free lunch. If you want something that's valuable, you have to pay the price. And here, potentially, this is extremely valuable because of the huge information content that you can access, but you have to pay the price in terms of the instrumentation that you need, in terms of the complexity of the analysis, but I think it'll be well worth it. There's plenty of examples that we already have, and there will be more to come.
Great, thank you so much, Klaus, and for providing us with such in-depth answers to some of those audience questions. So I'm afraid that's all we have time for in today's session. So any other questions that come in will now be addressed offline. So if you do have any other questions for Klaus, please do feel free to send them into webinars@bioanalysiszone.com. So I'd once again like to thank our wonderful presenter, Klaus, as well as you, our listeners, for your time and your questions. Don't forget to visit us at bioanalysiszone.com for more webinars and resources. Thank you again for attending, and we'll see you soon.
Thank you.